get started with transpiration. Now, all of you know that in the leaf, there are lots of processes that take place, such as photosynthesis, creation of oxygen, creation of water. So basically, carbon dioxide is used up by the leaves for the process of photosynthesis, and as a result, oxygen and water are given out as waste products of photosynthesis. Now, these two are gases, and gaseous exchange in the plants, it does not take place directly through the epidermis. It takes place through pores known as stomata. Now, why do you think the plant needs pores? It already has a skin or an epidermis. But you need to remember that on the upper layer of the leaf, there is a covering of cuticle, which is a thick, waxy covering, and cuticle is not permeable to gases. As a result, plants need to have pores or openings known as stomata. Now, when the stomata, they open up to let in carbon dioxide, inevitably water and oxygen get leaked out. And this loss of water in the form of water vapor, this loss of water is known as transpiration. So that is going to be the process that we will study. And you know that water, it actually comes from the roots and then it goes to the stem, and from the stem it goes to the leaves. And the material is carried through it is carried in the form of substances through special cells that are present in the tissue of xylem. So xylem is part of the vascular tissue of a plant, and that is responsible for the conduction of water and minerals from the roots till the leaves. Now, it's not a very simple process, and we need to go through different steps of that. That is what we'll be covering. But before going ahead to xylem and process in detail, let's learn a little bit more about stomata. So stomata are basically pores, and they are enclosed within two guard cells. So when you have dicot plants, you have kidney-shaped guard cells like this. So that is for dicots. And when you have monocot plants, they are dumbbell-shaped guard cells like this. So this is for monocots. Now these are guard cells, and around them you have the remaining plant tissue. Now the guard cells may also occasionally have chlorophyll in them. That means they can also take part in the process of photosynthesis. Alright, so these are the guard cells. So when the guard cells are open, they are very turgid, they are full of water, and they are ready to take in more water. So water is currently going in when these are open, and when they are closed, they become more flaccid because they are letting out the water. So that is the difference between appearance of the guard cells when they are open and when they are closed. Now coming back to this process of transpiration, the simple one word to describe how water and minerals get conducted from the roots in the upward direction against gravity till the leaves, a simple word is capillary. So the process is the capillary action. So capillary action is responsible for the conduction of water and minerals from roots to the leaves. Now in the next slide, we are going to discuss a special instrument that helps us to measure the rate of transpiration. And that instrument is known as a photometer. So I'll go ahead and draw a diagram for that. So this is what a photometer looks like. Over here you have a water beaker. And this over here, you can see this is the capillary tube which is extending from the water beaker to this container. This over here is a reservoir in case more water is needed in the container. So the tap can be adjusted to let in more water. And this is part of a green chute. So basically how the transpiration is measured is, now as transpiration and the evaporation of water goes on the leaf side, so it's going to pull up water, and over here will be an air bubble that will move towards this. So the distance that this air bubble moves as a result of the pressure by the leaf due to transpiration, That distance and the time in which the bubble covers that distance will give us an idea of the transpiration. So that is why we've put up a scale over here so that we can measure the distance the bubble moves. In case you want to push the bubble back, you will open this tap and let in the water, and the bubble will be pushed backwards. So that means the bubble can be reset for different plants or for different materials. Now, one thing over here about the chute is this chute is cut underwater and it is put into this container also underwater so that no air bubbles are formed and no air is leaked in. So the entire apparatus is completely airtight except for this one bubble. So that is how a photometer works. Now the next important thing that you need to know is how does water actually go through the way of xylem. So for that we need to know what is the structure of the xylem vascular tissue. So that is the next topic, the structure of xylem. So these are the two kinds of tissue or two kinds of cells that are present within xylem. These are known as tracheids. And these openings that you can see, these are pits. These are modified openings and they help to allow water movement between the cells. And one more thing, tracheids are hollow from inside. They do not carry any cytoplasm. And the second kind is vessels. Now these vessels have end walls that again have perforations in them to allow the water movement in an easy way. And these thickenings that you can see, these are rings of material outside these vessels. These are actually lignin. Lignin is a polymer and it helps to give strength and support to the xylem vessels so that it can support water upwards against the direction of gravity. Now, one more important difference between tracheids and vessels is that tracheids were there in the ancient flowering plants. The ancient flowering plants, the old ones, they had only the tracheids, not the vessels. So vessels are part of evolution. And the modern flowering plants, the modern ones, are the ones that we can find nowadays, they have only vessels. So the modern plants have vessels while the old ones, they had tracheids. Now the xylem structure is made in such a way that it can withstand low pressure also, and it is able to handle the cohesive property of water. That means water molecules, they tend to stick with each other because of the presence of bonds between them. And I already talked about the walls being thickened with lignin so that they become strong, and even when pressure is low, they do not collapse. Another thing that you need to know about the xylem vessels is that these are usually arranged end to end. That means on the top of each other, and the perforations between them, they help to transport the water. Now sometimes the cell wall between them, it breaks down and the cell contents also break down. So as a result, what you have is non-living cells without the cytoplasm. But non-living cells also need to allow water to be transported, but since water cannot actively flow, it is a passive process that needs to be done. So the flow of water through the xylem vessels is a passive process. And one more thing about the pressure that you need to know, that the pressure inside the xylem, is always less than the atmospheric pressure. That is one important reason why we need lignin to strengthen the xylem vessels. So this pressure difference plus the water traveling up against the action of gravity, and additionally the third factor, which is the cohesive property of water molecules. These three things are needed because that is the reason why xylem vessels need to be strong. Now about the cohesive property of water, if you draw up a water molecule, it looks like this, and it is polar because there is a slight negative charge on the oxygen and slight positive charges on the hydrogen atoms. So hydrogen atoms are going to attract other negatively charged oxygen atoms towards them, and similarly the cohesive property goes on and the water is easily pulled upwards through the xylem. So that is all that you need to know about photometer, what is transpiration, and how the structure of xylem helps in transpiration.